laboratory periods or systems to tackle before the semester ends. Okay, so for today, there are 6 specimens or organs under the lymphatic system. Again, credits to Junquierra Basic Histology Text and Atlas, where I got most of the reference from. Okay, so due credit to them by A.L. Mescher. So let's begin. So the first organ here you have is your thymus. Your thymus is a primary lymphoid organ and it functions for central tolerance, okay? Induction of central tolerance. So it will prevent autoimmunity, meaning your cells will attack its own cells. Alright, so they originate also from the third pair of pharyngeal pouches, or specifically in the endoderm region, okay? So that's just nice to know, because also in your manual, there was a question there where it originated. Okay, so nonetheless, third pair, I'll write it, of pharyngeal pouches in your endoderm. Okay, so also it's good to note that during puberty, there will be an involution of your thymus, meaning there will be a decrease in your thyroid mass, which will result to a decreased output of T cells. And again, they function for induction of central tolerance or the prevention of autoimmunity. So the parts you have to take note lang of, there are 4, okay? So you have your cortex, medulla, septa, and your trabeculae. So, and 5. And your hassles corpuscles. So we'll tackle that one by one. Okay, so here you have your septa. So septa meaning their spaces or septations. This is just spaces that will divide the organ to separate lobules. Okay, you have a septa here. So just these spaces, okay, they divide it to lobules. So division of the thymus. Next, you have your, what do we see here pa? The medulla. Medulla is usually a lighter stained area, okay? So if you compare it to the cortex here in the outer periphery, so it's much lighter. So here in your lymphoid organs, most organs here have either a medulla or cortex. Like for example, your lymph node also has same parts. So you really have to specifically delineate microscopically what makes a thymus different, for example, from the lymph node. So we'll try to do that today. So what makes a thymus unique is the presence of your HC or your hassles corpuscles. So your hassles corpuscles, these are aggregates of thymic epithelial cells, EC epithelial cells, okay? Located primarily in the medulla. Okay? So the medulla, again, this is a lighter area, okay? So if ever, I'll show you in the next slide, there are much more better presentations. This is how you can delineate a lymph node from a thymus. Because again, lymph node also has a medulla and cortex, and they are also subdivided, no? But this one is one of your answers. So here, this is taken from the laboratory. You also have trabeculae, okay? So we already discussed septa. We discussed hassles corpuscles. Your thymus also has a capsule, okay, and a covering that encloses it. Your trabeculae now are this one pointed in yellow, okay? So this one in green, encircled in green. Trabeculae are usually bands or columns of connective tissue. So this one here, okay, that is your trabeculae. You can see other parts of that later on. The space here is your septa, okay, dividing it to lobules. So this is the thymus. Take note of the appearance, okay? Erase it to make it better. Next. Sorry, not next, back. So, if ever lang, this is the appearance, okay? Or this is what's shown, for example, in an exam, you have to really have a quick overview check of what you're looking at. So, for example, this can be your guide, okay? This is located in a lighter area. This seems like an aggregate. No, it's a mass here. Lighter area means it's in the medulla. The mass is that this is a hassles corpuscle. Hassel's corpuscle has probably more septations or larger, okay, rather than your lymph node. So you answer thymus. So for thymus, please take note of what you should remember again. Your septa, trabeculae, your Hassel's corpuscle, and the medulla, which is lighter, your cortex also. You can't really appreciate yet lymphatic nodules here. Okay, so the cortex is the outer darkly basophilic area. So you have 5 to take note that this will give you a clue that it is your thymus. We'll proceed to the next one. So next is your palatine tonsils. Palatine tonsils is considered mouth. So mucus associated lymphatic tissue. There's quite a few to remember for palatine tonsils. Just remember that first, a tonsil is a large irregular mass of lymphoid tissue in the mucosa of the posterior oral cavity or nasopharynx. Okay, so palatine tonsils, what you should take note, because there are other types of tonsils, for example, lingual or pharyngeal tonsils. If you're talking about palatine tonsils, this is in the posterior end of the soft palate. Okay, aside from that, we'll tackle it one by one in the next. Okay, so this is what we'll focus on. Your palatine tonsils has crypts. This one in green. So crypts. What are crypts? These are invaginations, deep invaginations within the organ itself. Okay, usually a palatine tonsil in the textbook said that they have at least 10 to 20 crypts. Alright, next thing we have to take note is the lining epithelium. Remember that this is still in the near the mouth or the throat area. So the lining epithelium for this is stratified squamous epithelium. Okay, take note, stratified squamous epithelium. That's the next consideration to help you identify. The third consideration, now we will go to the lymphoid tissue. So this is what's characteristic now with most of the lymphoid tissues that's evident, except thymus. Okay, thymus, we didn't really see these packs of lymphoid tissue. And I believe you already encountered this also in your lymphoid connective tissue. Okay, this is just a repeat. So third consideration will be your lymphoid nodules, as you can see there. Okay, so fourth consideration would be that there can also be salivary corpuscles. Okay, so I'll show that in the next slide. Fourth is salivary corpuscles, and we'll define that also. Okay, so take note that in this slide, if you see, there are many, these dot-like structures already your lymphoid cells. Okay, this can be T cells or B cells here. And this is still within your squamous connective tissue, right? So it's still within the squamous area, the lining epithelium. But now, if you notice, it's already infiltrated with your lymphatic cells. So sometimes in some occasions it will be difficult to distinguish this if they are only shown this as a palatine tonsil sample, or this is what you see in the field. So ideally you have to really check still the overview. That's the technique here in histo. You see the overview, you have lymph nodes, crypts, and lining epithelium. So you'll answer, if your question is identify organ, palatine tonsils. So here, so salivary corpuscles. Salivary corpuscle, we didn't find one, but usually a salivary corpuscle can just be single or darkened dot-like areas here within the crypt. Okay, within the crypt. How do I change this to a different color? So for example, there's like a mass here, no? Because salivary corpuscle is defined as degenerating lymphocytes originating in the tonsils and passing into the saliva in the mouth. So sometimes, if ever in ana, this can be classified as a salivary corpuscle, and this can still help you in identifying that this is palatine tonsils. Okay, so aside from that, if this is the side, this is in the actual laboratory, you see here lymphoid nodules. So it takes practice also to identify that this is a nodule and another nodule. You see your lining epithelia here, even though it's not that nice, that's stratified squamous. Okay, and sometimes a salivary corpuscle can really give it off that this is palatine tonsils. So next, Peyer's patch. I'll try to run short, go through this fast since this was already taken up also in your, I already discussed this in Koan, your lymphoid connective tissue. So Peyer's patch is aggregates of lymphoid nodules in the submucosa or the mucosa layer of your ileum. Ileum, if identify the specimen, Peyer's patch of the ileum, okay? Or identify area pointed at Peyer's patch. Sorry, okay? If ever the question is identify the organ or specimen, you will answer ileum. If you ever identify the area pointed at, you will answer Peyer's patch. And how will you know that this is Peyer's patch? You look at your guiding structures. First is that the villi here, okay? The villi that usually has your goblet cells. If you view it in more, and let's change the color back to green. If you view it more, you will see goblet cells in this villi. So, here, this area, okay? So, what is it pointing at? What is the pointer pointing at? This is now your Peyer's patch, okay? Always look at the overview of the slide first. So, what else can we give it off that this is Peyer's patch? I think that's the best key to look at. No, you look at surrounding structures, goblet cells. So, also, just here, what makes this special is the presence of M cells. You can't really see this microscopically with a light microscope, but nonetheless, M cells, if ever we stack it up, so this is just kind of a short lecture that will relate with the M cells. This is your in the villi, okay? This is a simple columnar epithelium, and usually this can be a goblet cell on top. Your M cell will be in the middle, okay? It will be the middleman, and beneath your M cell is a intracellular pocket, as what was defined by the book. So the intracellular pocket now contains populations of your lymphocyte dendritic cells, okay, and dendritic cells. So lymphocytes and dendritic cells that leave the M cell pockets through the basement membrane. So here also there is a basement membrane, okay, meaning they will just pass out. The M cell will just be if this was a train station, the one where you or a bus station, the one where you input your card and then you can pass na. So nonetheless, the antigens from the lumen area on top will be passed on here, and later, since this is lymphocytes and dendritic cells, they will function to present the antigen to each other and then pass it on later to the B cell in order to make an antibody. And the antibody will counteract or neutralize or fight out the antigen. Okay? So, yeah. So here, it's very hard to appreciate this since this is an electron microscope. But the porous areas here, this is a villi man. So for example, there's a goblet cell here. It will help you understand better. Okay, so it will be taken in slowly, since remember columnar cells, they also absorb. And then the M cell, let's just say this is the M cell. It's very porous because it passes going upwards to the villi or the columnar epithelium and going downwards to your intracellular packets. So these sieve-like areas are how there will be exchange. Okay, so here's your basement membrane, and I believe this is the M cell. Okay, since M cells are still an epithelial cell. So here, this is a better presentation that will go on it all. Just take note, M cell gets the antigen. Where's the antigen here? The antigen is, I think, the green dots. Okay, yeah, ilalagay. So helper T cell will pass it on to your B lymphocyte. B lymphocyte later on will progress to becoming a plasma cell. Plasma cell will produce the antibodies, IgA, that will counteract or neutralize it, no, the antigens that's absorbed, and it will also be gone. So that's how the M cell plays a huge importance in your lymphatic system in your Peyer's patch. Next is vermiform appendix. This is very short. So the vermiform appendix is a short, small diameter that is a projection from the cecum. Okay. How do you identify this as vermiform appendix? First will be, your identifying feature will be your lymphatic nodules. Second is there is irregularity between the crypts, again invaginations, no, and intestinal glands or glands. Okay. And another giveaway to help you identify that this is appendix is there's a lumen. So see, you have a lumen here, no, in the center. Sometimes in the lumen there can be fecal material or microbial flora or bacteria. You see crypts here, and sometimes intestinal glands. Okay, within the just below the lumen. It's always round in structure. This is still a mucus-associated lymphoid tissue, similar with your palatine tonsils and your Peyer's patch. Okay, so again, see, you have another lumen here. It's very evident your lymphatic nodules here that's stained blue-purple, and then you also have your intestinal glands and your crypts here. Okay, very nice specimen, very nice. So crypts. So that's all your giveaway. Here, this is a slide from the laboratory. You see lymphatic nodules here. So lymphatic nodules, you see your glands and crypts. Glands plus crypts. And there's a lumen pudged. So that's immediately identify the specimen, vermiform appendix. Identify structure pointed at lymphatic nodule. Okay, so we'll go to the second to the last, which is your lymph nodes. Your lymph nodes, this is a bean-shaped capsulated structure distributed throughout the body. The main function of the lymph node is that it is an inline filter. Okay, inline filters of lymph that prevent against the spread of microorganisms. Prevention of spread of microorganisms. So again, they provide an enclosed environment for antigen presentation, where in your dendritic cells, your T cells will present an antigen found in the lymph to B cells. B cells activated later on or develop the plasma cells antibody produced, or sometimes it can just be a natural killer cell, or it will promote cytokines that will attract macrophages or other first or second line leukocytes that will destroy out the antigen or the microbacterium. Okay, so you have parts. It's subdivided particularly to three regions. First, the first part here is your cortex. Second is your paracortex, and the third is your medulla. So note that in your paracortex, there's no precise boundaries, no, but it can be distinguished by the lack of lymphatic nodules, which is only found in your cortex. Okay, so the cortex also has a subcapsular sinus. So subcapsular, meaning it's below the capsulated areas. Sinus, it's a space. Okay, so this is a subcapsular sinus. Aside from that, the cortex will also hold, again, your lymphatic nodules. The lighter area in your lymphatic nodules is the germinal center. Okay, the darker area is your primary nodules. Primary lymphatic nodules. Germinal center is also known as your secondary lymphatic nodules. Okay, you can also still see trabeculae in your lymph node. So what is trabeculae again? These are bands or columns of connective tissue. Later on, if you go to the medulla najud, you will see cords. The cords are these dense cord-like basophilic structures, and these are masses of lymphoid tissue. No? Masses najud. The sinuses, so if you're talking about medullary sinus, these are dilated spaces. So again, the word sinus immediately come into your mind, space. These are dilated spaces lined by discontinuous endothelium that separates from medullary cords. White area, if that's what's pointed at, if identify structure pointed at, medullary sinus. Okay? So here, capsule, subcapsular space. So below, subcapsular space, or sinus. So lymphatic nodule present. So if ever there's a question, identify the area pointed at or the region pointed at. You will answer cortex. Identify the structure pointed at, lymphatic nodule. Okay. There's no germinal area present, so this is primary lymphatic nodule. Identify the structure. So hey, there's no Hassel corpuscles, no, in the cortical area, subcapsular space, and then so you'll answer lymph node. Okay, so see, if you notice, unlike the thymus, there's many spaces. Or invaginate jud mo, it's really evident, but here not so much in lymph node. Okay, so here, this is the ones taken from the slide. Alright, so again, subcapsular sinus. So sorry if ever it's repeating again and again. So I just want to show you the difference from the actual lab and from the textbook. So it can really help and supplement your learning, especially if you decide to pursue medicine or in the other microscopic studies in third year. So you will be acquainted microscopically na. So you have your lymphoid follicles or your primary nodules. So the germinal center is also known as your secondary lymphoid nodules or lymph nodules. Cortical sinus present because these are spaces now still within the cortex. It's in your cortex that you find your nodules. Paracortex, wala na. So this is paracortex, cortex. So you go deeper pa jud in the medullary region or area or layer, you will see your cords, mass like lymphatic structures, then the sinus, which is your discontinuous dilated spaces. Okay, that's that. Last specimen is your spleen. Spleen is located in the lower left upper quadrant. It has the largest accumulation of lymphoid tissue. So again, largest accumulation of lymphoid tissue. Okay, and then this is the only organ that's involved in blood filtration. So it has a role in your blood-borne antigens. So unlike the lymph node, the thymus, they don't have a direct role in red blood cells or blood per se. Okay, this one it has. Also, your spleen will also still function for the production of antibodies, just like others, no? By the way antigens are presented, T lymphocytes, and then later on to B, to plasma cells, to antibodies. Okay, they still do that. And what makes this unique is also its parts. So it's subdivided into two parts. There's a red pulp and a white pulp. Okay, so we'll discuss that one by one. So the white pulp of the spleen. See, I placed the definition again, credits to Junquierra. So it will be easier to understand because even before I get confused, it's called white pulp, but the color is actually purple. Red pulp, so it's mostly red in appearance. No? So let's start with your white pulp. The white pulp is 20%, no? And the red pulp comprises 80%. Red pulp. So in your white pulp, this houses your lymphoid tissue, okay? Lymphoid tissue or your lymphoid nodules. Aside from that, there's also the presence now of your pulse. So what is your pulse? This is your periarteriolar lymphoid sheath. So look down below. So I'll make this red lang. Once we talk about vasculature first. The trabecular artery will branch off to a central arteriole, okay, in your microcirculation of your spleen. So once it becomes a central arteriole here, central arterioles are usually sheathed. So they are sheathed with this periarteriolar lymphoid sheath. Usually there's lymphocytes also, no? Within that sheath that sandwiches the central arteriole that passes to your white pulp. So white pulp, this is so this germinal center here, if you can see this, if this is in this diagram, it is your lymphoid tissues, okay, lymphoid nodules. It will pass there. Later on, it will branch out again now to your arterioles. So peninsular arterioles later on, and then finally it will branch out to capillaries. And with capillaries, this from artery circulation, it will go to a venous circulation, the blue one. Okay, so that's just how it is. That what you can see now in your white pulp therefore is a central arteriole. Remember we talked about arterioles in your cardiovascular system. There's always a lumen, there's less number of layers and elastic lamellae that is admixed with your muscular layers. So here now, so immediately if you see an artery, if you see a lymphoid mass, and you're asked identify the layer or identify the area, so immediately you will answer white pulp, okay? So that's just what I want to emphasize, that what you see in what areas and what, okay? So I won't make it that long. So the red pulp now is a different region, okay? So we discussed the white pulp now. Even you can see it here, no? That this is a central arteriole. Now we'll discuss the red pulp. So the red pulp mainly consists of your splenic cords of Billroth, splenic cords of Billroth, and your splenic sinusoids. So your white pulp consists of, I'll just write it here also, your lymphatic nodules and your periarteriolar lymphoid sheath with your central arteriole. Man, it can't be seen. Anyways, I hope you understand it through the audio. So the last part, what are splenic cords of Billroth? So these are a network of reticular cells and fibers with T and B lymphocytes, macrophages, leukocytes, and RBCs. So within these cords, there's a party of many other cells. The lymphocytes, the L with a cross in it means lymphocytes, okay? B lymphocytes, macrophages. You also have what else? Leukocytes or WBCs and your RBCs. So they're running within these cords, okay? So these are Billroth cords. Billroth cords. These cord-like structures. I hope you're very imaginative that you can admire and see it. Okay? Billroth cords. So that's it. That's what you can see in the red pulp. And aside from that, you can also see your splenic sinusoids. So your splenic sinusoids are lined by these cells, and I don't think you can see this, or maybe you can since it's digital, but usually in lab-wise it's very hard to identify it, okay? But if you really zoom to a higher magnification like this, that's possible. So these are stave cells. What are stave cells? So these cells are endothelial cells that line your splenic sinusoids. What do they function for? They allow separation of healthy RBCs from defective or old RBCs, meaning the healthy RBCs and even other cells, like for example platelets, white blood cells, they can diapedes, or they can pass through. They're allowed to pass through to enter circulation again. But the defective red blood cells, they are taken and engulfed or broken down. That's why they call the spleen as the RBC graveyard by the spleen. Okay, and that's why they function, particularly for blood filtration. This is where your stave cells, stave, S-T-A-V-E, these are endothelial cells that function for blood filtration. They won't, old RBCs not allowed to pass through and re-enter circulation, taken in, engulfed, broken down in spleen. Okay, so again, these are the two things you can see in your this is under splenic sinusoids. Okay, splenic sinusoids in your red pulp. Okay, that's all. Thank you. Sorry, last na jud. So here, this is seen in the lab. Okay, so Billroth cords seen here, very claro. So the cell party, splenic sinusoids. So see, this is what I said. It's very hard to see your stave cells, but the splenic sinusoids na lang are the white area spaces. Okay, and then the white pulp, and here you can see a very evident. I'll erase it so you can appreciate it. A sheath central arteriole lymphatic nodules. Very easy giveaway that this is definitely the spleen if you're asked to identify the organ, which is spleen. Okay, thank you very much, and that's all for this week. Hope this supplemented your learnings. Thank you. God bless. Stay safe.